remains in the arterioles or the capillaries is going to be the blood, okay? And then I just have a note down here just so we've got an idea of what we're dealing with. You're talking about roughly 45 gallons or 180 liters of blood being filtered through into Bowman's capsules of all of these different nephrons that occur within the kidney. Okay? Why I've got everything kind of scattered throughout my notes. Need to move down through there. Okay? All right. In order to get to that 45 gallons of blood to filter, you have basically 1 million of these nephrons, okay, associated with your two kidneys, about 1 million nephrons. And the nephron itself, the capsule all the way through the tubule to the collecting duct, you're talking about 80 kilometers of tubule. So there's a lot of surface area in this thing that helps with all of this, right? Reabsorption, secretion, excretion, and filtration. Okay? All right. So far, so good. So squeeze the blood, filter, it gets collected into this capsule. Should draw a tube out here. So the filtrate is being collected in Bowman's capsule, and then the filtrate would move from the capsule into the proximal convoluted tubule. Okay? All right? Okay? All right. So if we keep with this capillary language, that would be the glomerular capillary. And then we would have capillary associated with these tubules, both the proximal and distal convoluted tubules, as well as the descending and ascending limb. So we're just going to start to break these apart language-wise. You have the glomerular capillaries, or the glomerular arterioles, doesn't matter to me. And then here you've got what are called the peritubular capillaries, because these capillaries are around the proximal and distal convoluted tubules. So peritubule, peri meaning around, and tubular, those parts of the nephron. That's peritubular capillary. And then down here, the capillaries where the artery system comes together with the venous system, which for me feels like a true capillary. This portion here is called the vasa recta. So I could kind of draw this a little bit. You've got Bowman's capsule, and into the collecting duct, that's the descending and ascending limb, right? The proximal convoluted tubule and the distal convoluted tubule and the collecting duct, and then this part here is Bowman's capsule. So you have the glomerulus here, then you would have peritubular capillary here, and vasa recta here. But it's all part of the same system. It's just some biologists enjoying kind of breaking these into pieces with new terminology. The reason for that is these different portions of this capillary system are doing different things, so it kind of helps to break them out language-wise, because there's different functionality and different physiology. Right, the anatomy goes with the physiology. Right. All right, so we've moved that filtrate and we pass it to the proximal tubule. It's called the convoluted tubule because it's convoluted. It folds back and forth across itself. And then the proximal because it's closer to the front end of the nephron, or closer to Bowman's capsule. So the proximal convoluted tubule. Okay? So what's in the proximal convoluted tubule now is the filtrate. Right? So the filtrate is moving in. And then we're going to move through this proximal convoluted tubule part of the nephron. Okay? All right, so we're going to take these in pieces and then try to put it together afterwards. But in the proximal convoluted tubule, the filtrate should contain a bunch of stuff. That should be a lot of water, because there's water in the circulatory system, right? I think we said the largest component of circulatory system blood was that water component. And then associated with that water should be anything that was small enough to fit through those glomerular fenestrations and slits that are in those podocytes. So the filtrate now includes a bunch of stuff. It should include all kinds of salts, right? Sodium ions, chloride ions, as well as potassium, calcium, manganese, magnesium, right? All these ions. We've also got a fair amount of bicarbonate that's moving through the circulatory system. That's because of what's happening with the respiratory system. So we have a lot of bicarbonate ions that are moving around as well. And because we've got bicarbonate, we also have that hydrogen ion, right? Because CO2 in water yields carbonic acid, which gives us bicarbonate and hydrogen ions. So we have hydrogen ions in here as well. Additionally, we have urea, which is that ammonia has been converted to the urea. That's the nitrogenous waste. We want to get rid of this stuff, and also at the same time balance out as much of this other stuff as we can. And then there should be some biological molecules, right? Carbohydrates in the monomer forms, individual amino acids. Those should also move through those fenestrated glomerulus cells, as well as the podocytes, and then also some drugs. These would be things that are have been worked on by the liver, and instead of moving it from the liver to the digestive system, right, through that duct that moves from the liver to the small intestine, some of this stuff can be detoxified by making it water soluble. Then the liver can dump it into the circulatory system and let the kidney worry about trying to get rid of it. Okay, so in your other classes, you'll talk about the liver kind of having two pathways to deal with toxins. One is to move it to the digestive system, and the other, in the case of the water-soluble molecules, get rid of them by having the kidney deal with them. So that's why this says some drugs here. Okay, so if we look up at the top here, the proximal tubule has associated with it these red arrows and these kind of lilac arrows, right, purple arrows. The red arrows represent active transport, which means there's an ATP cost to moving sodium chloride out of the filtrate and back into the body. And nutrients like glucose and amino acids, we're going to have to expend energy to move those back into the circulatory system. And then here, hydrogen ions can be moved into the proximal convoluted tubule, again through active transport. So just on the active transport side, we have two processes occurring simultaneously. There's reabsorption, salt, nutrients, and there's secretion, which would be hydrogen ions. Okay? All right, we also have some passive transport occurring. The passive transport occurs with bicarbonate, potassium ions, and water, as well as NH3. Now, NH3 is ammonia, and if we have the ammonia plus hydrogen ions, that should give us a bunch of ammonium, which is this one. Okay? So we're taking ammonia from the surrounding part here, in this case from the cortex. That ammonia is passively transporting, moving down its concentration gradient into the filtrate. That would be secretion. even though it's passive. And then that's combining with the hydrogen ions that were actively transported into the proximal convoluted tubule to form a bunch of this ammonium. Okay? Now, the water here is moving passively. Like I mentioned before, I'll mention it again. Organisms don't actively transport water. The reason for that, there's a lot of reasons, but the idea is water tends to go where it wants to go. It has properties of both polar and nonpolar depending on where those electrons are between the hydrogen and oxygen. So water can kind of move around. It moves through any protein channel that moves anions or cations. Water can move through those. It moves through aquaporins. There's a component of water, the water in cells that can move across membranes through osmosis. So it's hard to control the movement of water by moving water. On the other hand, you can control water by moving things that water likes to associate with. So if we transport out a bunch of sodium and the nutrients are acting as osmotically active agents, and we're moving bicarbonate and potassium ions passively out of the proximal convoluted tubule, you've moved a lot of ions and other osmotically active substances out. Water through osmosis will follow those. So in this proximal convoluted tubule, we're playing games initially to move water out, right, back out of the proximal convoluted tubule back into the body. Again, I think I have some numbers here. Yeah, you're going to dump in about 45 gallons are going to be processed per day by these nephrons. But at the end of this process, what's going to be released is this urine. A day, that's about 1.5 liters. So 45 gallons is 180 liters of filtrate down to 1.5 liters of urine. Well, most of that difference is water. So we're moving this water back out of the filtrate to keep it in the body. Okay? So that's that proximal convoluted tubule. We're adjusting osmotic balance and we're adjusting water balance. Okay? All right. Okay. The other thing about the proximal convoluted tubules is its design is kind of interesting. So these tubules, if we took a closer look at these and saw how they were organized, there's actually more surface area on the inside than there is on the outside. So if this represents the kind of the outside of the tubule, the inside of the tubule has more surface area. Now the additional surface area on the inside relative to the outside means if I actively transport something across, right, using this additional surface area, that means leakage would allow less of it back in, because what helps to limit the movement of stuff back into the cell is the surface area of that tubule. So if there's more surface area on the inside than the outside, that means it's easier for me to transport stuff in to out. It's more difficult for stuff to move out to in. So what we're doing is actively transporting in a way that helps us, and we're reducing the amount of passive transport that would undo what we're trying to do actively. Okay? All right. Okay. All right. That takes us now here, after we've made some initial adjustments to the filtrate, doing both reabsorption and secretion, that takes us now to this descending loop of Henle. Now, in the descending loop of Henle, as we move deeper into the medulla, okay, as we move deeper into the medulla, the salt concentration increases. Why that is, let's get all the way through the nephron first, and then we can put it all together. But for right now, the environment in this medulla has been maintained in such a way that we have higher salt concentrations down toward the inner medulla and lower, a little higher in the outer than the inner, but certainly more concentrated salts here than here. So whatever water was moving out of the proximal tubule, we're going to get more water to move out of the descending limb of the loop of Henle because this area is more concentrated. It's more hyperosmotic than the cortex is. Okay, so again, manipulating salt balance outside the tubule in order to get water to go in a certain direction. So here, the water is moving out passively. Okay? Now, the weird part about the descending limb of the loop of Henle is the tubule itself has been designed to allow for the movement of water, but salts aren't allowed out. So you could imagine the construction of this tubule in such a way that salts get trapped in the tubule, salts can't leave, but water is pulled out very easily because we facilitated the movement of this water through facilitated diffusion. We're not allowing the salts out. So in the descending loop, water can move, but salts can't. Water moves from a high concentration in the filtrate to a low concentration in the medulla. And then you can imagine in those birds and mammals where these loops descend very deeply into the medulla, right, these juxtamedullary nephrons, that gives us more of a chance to adjust water balance because we've got more surface area across which to move the water. If these nephrons didn't penetrate very far, right, I had a cortical nephron where the loop terminated here, that would not be very much distance across the loop of Henle to make these water adjustments. By driving it deeper in, I have a longer distance across which I can make these fine adjustments to water. Okay? All right. The filtrate is now moving up through the ascending limb of Henle. And as that filtrate moves